Welcome to The Explainer. Today, we're digging into a phase 2 trial that asks a really critical question. Can we dose oxaliplatin smarter? This study, called LEANOX, offers a new strategy to tackle one of the most challenging side effects we face in GI oncology, peripheral neuropathy. Okay, let's jump right in. We're going to follow a classic journal club format to really get to the heart of this paper. We'll start with the problem it's trying to solve and finish up with what this all means for the patients we see every day in the clinic. So first up, let's set the stage a little. Why was this study even necessary? Let's talk about the clinical challenge that comes with using oxaliplatin. The scale of this problem is huge. We're talking about almost half of our patients on adjuvant FOLFOX developing some level of neuropathy. This isn't some rare side effect, it's a daily clinical reality that really impacts treatment and, of course, quality of life. And OIPN isn't just, some pins and needles. As we all know, it's a chronic, dose-limiting toxicity. It's the very reason we're often forced to cut back or even stop a key drug in a curative intent regimen, and we're always left wondering, did I just compromise their outcome? So this brings us to the core issue the LEANOX trial is trying to tackle. For years, we've been using a dosing metric from the early 20th century, body surface area, for a drug with a pretty narrow therapeutic window. It just completely ignores whether a patient's weight is muscle or fat, which could be absolutely critical for how they metabolize and clear the drug. And that brings us to the trial itself. The LEANOX investigators came up with a hypothesis that there's a more personalized, a more precise way to dose this drug. Their whole hypothesis centered on a new metric, lean body mass, or LBM. The idea is that LBM, which really reflects a patient's muscle mass, is a much better predictor of drug tolerance than BSA. And you know the best part? It can be calculated right from the routine pre-treatment CT scans we're already ordering for our patients. So here are the quick facts on the study. It's a phase 2, multi-center, randomized trial out of France. They looked at 160 patients with resected stage 3 colon cancer who were getting adjuvant FOLFOX. And the central question is simple and super relevant. Can this new dosing strategy actually reduce clinically significant neuropathy? Now, this right here is the key comparison. The study first identified patients with sarcopenia, so low muscle mass, and then randomized them. Arm 2, the control group, got the standard dose we always use based on BSA. Arm 3, the intervention group, got a dose calculated based on their lean body mass. This head-to-head comparison in this really vulnerable population is exactly what makes this trial so compelling. And the primary endpoint they chose was very pragmatic. They looked at the percentage of patients who avoided getting grade 2 or higher neuropathy within the first 3 months of treatment. This is that critical time frame where toxicity often derails therapy, right? And grade 2 is where it starts to impact a patient's day-to-day life. Okay, let's get to the good stuff, the results. Did this personalized dosing strategy actually work? Well, it did. The primary endpoint was met, and the result is pretty significant clinically. In the standard dosing arm, only about 42% of patients avoided significant neuropathy. But in the LBM-dosed arm, that number jumped to over 67%. That's a 25% absolute improvement, and it was statistically significant with a p-value of 0.01. And what's really convincing is that the secondary endpoints all pointed in the exact same direction. Patients in the LBM arm took more than twice as long to develop significant neuropathy. We're talking 5.7 months versus just 2.3. They needed way fewer dose reductions, and their neuropathy-free survival was a lot better. It tells a very, very consistent story. This, of course, is the million-dollar question anytime we talk about de-escalating therapy. It's great that we reduced the dose and we reduced toxicity, but did we hurt our patients' chances of a cure? Well, reassuringly, the data showed no evidence of harm. After a median follow-up of nearly 39 months, there was absolutely no difference in relapse-free survival. The hazard ratio was essentially 1.0. And the overall survival data tells the exact same story. Again, no statistical difference between the two arms. So this provides some really strong reassurance that this dose personalization strategy did not seem to compromise oncological outcomes, at least in this trial. So if we look at quality of life, the patient-reported outcomes on that specific neuropathy questionnaire, the QLQ-CIPN-20, were better in the LBM arm, which just confirms the primary endpoint. Overall toxicity was pretty comparable, but it is important to point out one signal. There was a higher rate of grade 3 or higher neutropenia in the LBM dosed arm. It's definitely something to be aware of. So what does this all mean for us in the clinic? With these results in hand, let's talk about the limitations and the next steps before we think about just adopting this strategy tomorrow. Of course, we have to be critical here. This study was done in a primarily Caucasian population, and it wasn't blinded. And while that survival data is really reassuring, we have to remember this was a phase 2 trial. It wasn't powered to definitively prove non-inferiority. And finally, there's the practical challenge. How do we implement LBM calculations in a busy clinic? But even with all those caveats, the editorial that came with the paper is actually quite positive. Dr. Koh from JCO suggests that this approach is, quote, appropriate to consider, especially for our patients with sarcopenia. That's a pretty strong signal that these findings are being taken seriously. This really points to where this strategy could have its biggest impact. I want you to think about the patient with sarcopenic obesity. You know the one, low muscle mass but a high BMI. Their BSA-based dose can be sky high, and these are often the very patients who run into trouble with toxicity. For them, LBM dosing could be a real game changer. Okay, so let's wrap this up and distill it all down to the clinical bottom line. What are the key takeaways you need to know from this LEANOX explainer? Here are the 3 main things to remember. First, dosing oxaliplatin based on lean body mass significantly, and I mean significantly, cuts down on debilitating neuropathy. Second, this reduction in toxicity did not seem to come at the cost of efficacy in the study. And third, this trial gives us a data-driven blueprint for how to start personalizing chemotherapy for a really vulnerable group of our patients. The LEANOX trial gives us some really compelling evidence, and it leaves us with this provocative question for the entire field. With software making this more and more accessible, are we entering an era where CT-based body composition analysis actually becomes a new standard of care for chemotherapy dosing? That's all for this explainer.